Y antes de comenzar esto, quiero hacer dos pequeños anuncios que este congreso, resaltar, en gran medida ha sido posible que se lleve a cabo con eh, la organización y los detalles que todos podemos apreciar gracias al aporte de la SMBI, que ha estado apoyando muchísimo todas estas actividades en varias maneras, principalmente también económica. Y eso es para resaltarlo y estamos muy agradecidos. Y también recordarles que esto está siendo transmitido de manera simultánea a la sede en Guatemala. Por lo tanto, en el momento de las preguntas vamos a tratar de intercalar, si es que hay preguntas en Guatemala, algunas de acá y algunas de la audiencia que está de manera virtual. No me quiero olvidar de nada, así que voy a leer. Dan es profesor del Departamento de Ecología y Biología Evolutiva y es curador de la colección de la herpetología en la Universidad de Michigan desde el año 2012. Y sus principales temas de investigación son macroevolución en vertebrados, principalmente en lagartos, especiación, métodos filogenéticos comparados, biología evolutiva, y también trabaja en el desarrollo de paquetes analíticos en R relacionados a análisis macroevolutivos y filogenéticos. Es autor, como se imaginarán, de muchas publicaciones, más de 160, y actualmente hemos disfrutado de su presencia en Argentina y que nos pueda brindar esta conferencia. Así que con esto le damos la bienvenida y le agradecemos su participación.
Okay. Thank you very much for that introduction, and thank you very much to the conference organizers for inviting me to talk about my work here. Can everyone hear me okay? This is good. Okay. So I unfortunately pulled a little bit of hay una expresión in English, bait and switch, or false advertising, in that the title of my talk is in Spanish. And unfortunately, I am going to give my talk in English so that more people will actually understand it than if I try to give it in Spanish. So I apologize for that. But before I start, I want to just say one thing that might explain a little bit of some interactions you might have with me after my talk or before. And that is that, and you might not have noticed this, but I have severe to profound hearing loss. So I have more than 90 decibelios in each ear. And might not notice this, but it makes it very difficult for me to have conversation if there's a lot of background noise, like in poster sessions or restaurants or other things. Yo leo labias en inglés, pero necesito practicar mucho más en español antes de que poder leerlas en español. Y por eso es muy difícil para entender español cuando hay ruido en el ambiente. So if you don't, si no estoy respondiendo a lo que decís, la explicación más probable es que estoy teniendo dificultades para oírte. So, and I want to start by thanking a number of folks whose work I will discuss in my presentation. And these include some former postdocs and students of mine, Sonal Singhal, Ivan Pratas, Pascal Title, Mike Grundler, Mateus Januario, and many others, although those five are going to be featured prominently in some of the things that I'll talk about. And I also want to say that some of the empirical things that I'll talk about all rely very heavily on specimens that were from natural history collections. In some cases, these are things that we have collected ourselves in my lab, but this is all impossible work without the work of literally hundreds of museum biologists around the world who have put specimens in collections. Those are some of the museums that we have relied most heavily on for some of our own work. And I also want to thank, especially for the last 6 months, we have been sabbatical visitors in Puerto Madryn at CONICET. So I want to give a special thanks to Mariana Mirando and Luciano Avila for hosting my wife, Allison, and I as sabbatical visitors. So it was an incredible place to spend 6 months. And so, some of the ideas that I'll talk about today were some things that I made progress in thinking about while I was hosted there at CENPAS. So thank you very much for hosting us in Puerto Madryn. Okay, so I want to start today with this very general question of what is macroevolution? Because macroevolution is a lot of different things to a lot of people. And I think it's worth thinking about the history of what macroevolution is. And to start, I'm going to give this example, and I'll come back to this a few times. So these are representatives of major groups of Australian squamate reptiles. So this is a group of organisms that we work on empirically in my lab. You can see there's a lot of diversity of body form. You have big things, you have small things, you have legless things, you have spiny things, you have smooth things, and so on. And so each of these body types here is kind of representative of a different major clade of squamate reptile that lives in Australia. So these are lizards and snakes. And classically, one of the ways people thought about macroevolution during the early modern synthesis in evolutionary biology was to think about the fact that you have these patterns of variation that people could see within species. So systematists started putting specimens in museums, and people, starting around Darwin's time, started to understand intraspecific patterns of variation. And so people could see that, you could look at differences between populations of the same species, and you'd see there's some morphological differences between those things. Now, this graph is nothing that you will find in this literature, but it's sort of my representation of how people were thinking about it at the time. But people were really struck by what they saw as patterns between species, where there seemed to be a clear break between the qualitative kinds of differences you saw between species and the kinds of variation that they would see between populations of what were obviously the same thing. And Richard Goldschmidt was a geneticist who was a big advocate of this idea that macroevolution was this break between within species variation and differences between species in terms of morphology. But many paleontologists and others also thought about this even at higher levels. So they thought, well, you actually saw within groups, like within snakes, there are differences between snakes, but they're very different from the kinds of differences that separate snakes from other groups of lizards. And so it doesn't necessarily have to be the within species, between species level, but it's basically within some major taxon versus between taxon. And so, this led to a lot of, speculation about the ways in which processes that we could see happening were or were not adequate or sufficient to explain these big gaps. And so even Goldschmidt said, they made this famous quote, where the facts of microevolution, see with genetic mechanisms, were not sufficient for an understanding of macroevolution. And so macroevolution in its classical sense was kind of addressed, this morphological variation between groups was addressed by many of the works of the modern synthesis. The major point in some ways of these works, by Simpson or Dobzhansky or Meyer or Huxley, was really to understand, show how the origin of species and the evolution of morphological differences between groups were sufficiently accounted for by what we knew at the time in terms of genetic mechanisms. And so that in some ways is a major point of these works here. And so macroevolution at this time is really originally about how major morphological forms or higher taxa originate, or how those morphological novelties or differences that separate groups, ranging from between species differences to between big clade differences, originate. And I think when I look at evolutionary biology over the past 15 years in particular, I think we've made an enormous amount of progress in answering that basic question. These are just a sample of papers over the last few years that have looked at how the mutational process, the multivariate mutational process, what we think of as sort of the M matrix, or standing patterns of genetic variation, or covariation, what we think of as the G matrix, can predict larger scale patterns of phenotypic macroevolution. And while we have a lot more work to do in this area, I think these papers represent the fact that we are making progress in this particular area. And so this is just another example, a really nice paper by Joel McLaughlin looking at how patterns of genetic variation predict broader scale patterns of between species divergence within anolis lizards. And so traits in this case with more intraspecific genetic variants differ more greatly in terms of their between species patterns of divergence. And so, again, macroevolution here is thinking about the problem of how major morphological forms originate. What is almost entirely missing from this literature in the early modern synthesis and this original conception of macroevolution is how this relates to broader patterns of species diversity. And if you think about these clades of Australian lizards and snakes, for example, these are the species diversities of each of these groups. So you've got some groups, like up in the upper right, sphenomorphine skinks with lots of species, and other groups like some geckos with relatively few species. And so the variation in species diversity among groups was largely neglected. And so post-1960s, macroevolution has really started to include this idea of biodiversity dynamics as well as morphological evolution. And this is really about how speciation and extinction affect species diversity through time and in space and among clades. And there's sort of a whole nother story I could tell about how and why I think macroevolution came to encompass species diversity. I'm not going to really go there, but we're going to talk about briefly what some of these patterns are. So this is the distribution of species richness among clades of Australian squamate reptiles. These are lineages that colonized Australia and underwent some degree of diversification within Australia. So again, you see some groups like these sphenomorphine skinks on the left here that have lots of species. And then you've got this long tail of clades that have things that have basically not diversified at all, like naturescene snakes, which colonized Australia and didn't do any diversification, even though they've probably been there for a couple of million years and have had some time to do so. So what explains this particular pattern that we see in terms of species diversity by clade? We could look at the same thing for any region on Earth and any group of organisms. See, it's at higher level scales as well. This is the distribution of species diversity across major family level clades of squamate reptiles. So you can see there's just 3 clades really dominate total species diversity at that taxonomic level within squamates: geckos, skinks, and colubroid snakes, which just have a tremendous number of species, especially here in South America. Likewise, another part of biodiversity dynamics is thinking about species diversity through time. So this is alpha diversity in terrestrial plant diversity from the fossil record. And what you see here is that, it's kind of there's a number of things to look at here, but this pattern of changing diversity or not changing diversity through time is another facet of biodiversity dynamics. Here's another thing. This is this is the relationship. This is this is brachiopod and clam diversity in the fossil record. You see brachiopods. This is the dashed line there is the Permian Triassic extinction, and then after that point, clams basically start becoming more diverse, and brachiopods stay undiverse. Another sort of pattern that we are interested in within biodiversity dynamics. And I will say this figure is from this paper by Gould and Callaway, Clams and Brachiopods, Ships That Pass in the Night, which is in my top 5 favorite papers in evolutionary biology. If you haven't read it, I highly recommend it. And a final dimension to biodiversity dynamics to me is spatial patterns of diversity. So this is species richness varying across the surface of the Earth. So this is the distribution of species richness for marine fishes. So you can see that there's a huge peak of fish diversity in the tropical Indo-Pacific. And of course, this pattern is something that we see all over the place in every kind of geographic setting. The most famous sort of iconic pattern is the latitudinal diversity gradient. And I like this figure because it's about snake diversity. It's showing how snake diversity declines from northern Argentina in Misiones and Corrientes down to Santa Cruz and Tierra del Fuego. And it's also this, I think, figure 4. So it's the 4th figure in the first paper, to my knowledge, to concretely illustrate the latitudinal diversity gradient. And it's the paper from Fisher from 1960. And so again, this is another spatial pattern of species diversity. The other cool thing about this is it's about snakes. So, all right. So a little bit of background for my talk, and that's when I talk about speciation rates and extinction rates. So we're going to think about speciation rates, or the per lineage rate at which a species splits into new species. The extinction rate, I will sometimes label these things S and E. So if you see S on a figure, it's the speciation rate. If you see E, it's the extinction rate. And then sometimes we'll talk about this thing called the net diversification rate, which is speciation minus extinction. It's the net balance between these things. It's sort of the net rate at which species are building up or being lost from a clade or a region. And I'm not going to talk at all about methods in this talk. The only thing you need to know is that we have methods that exist for doing things like estimating rates from phylogenies, from the fossil record, and potentially from other sources of information. And so these methods can do things like take a time-calibrated phylogenetic tree like you have here and turn it into, with help from some math and some computational infrastructure, we can turn it into things like this, where this is a tree where I've now plotted the branches or colored the branches by their estimated instantaneous rate at which lineages are being popped out due to speciation along that tree. So just sort of a heat map of where speciation is fast, where speciation is slow. So my talk is focused on, I'm going to break my talk into 3 general parts. First thing I'm going to do is talk about a little bit of a personal history and critical assessment of biodiversity dynamics as a field as it stands now. Then I'm going to talk about an equilibrium model for biodiversity, and then I'm going to talk briefly about some evidence and some implications. So I'm going to make a set of controversial assertions here. Well, I don't think they're controversial, but other people might think they're controversial. First of all, I think that things that we think should predict speciation in a broader sense have consistently failed to do so. To me, this is one of the biggest findings in macroevolution in the past 20 years. And to understand what I mean, I'm going to give you a little bit of background about controls on the speciation process. And so I'm going to focus on things that should, from a microevolutionary perspective, control the dynamics of speciation at the macroevolutionary scale. And so our general classification that we work with is based on some ideas from Ernst Mayer's 1963 book, where he really talks about speciation as successful speciation as being a product of a number of distinct controls. And the first control he talks about, which he devotes the most time and space to, is really the formation of isolated populations, which in his case is heavily focused on geographic isolation. So ultimately, this has to happen to get successful speciation. But there are other ingredients as well, including things like the evolution of reproductive isolation as an intrinsic trait between populations. And that was obviously something that other folks from the modern synthesis thought a lot about, like Dubchansky's book is almost entirely about the reproductive isolation between populations. And Meyer talks a little bit about ecological differentiation between populations and what that means for population persistence as well. Now, I'm not going to go into details, but we can just assume for now that there are multiple things that have to happen to get successful speciation. And one of the things that we've been good at doing, I think, in the last 15 years is figuring out ways. We have a lot of challenges remaining, but we've been good at figuring out ways to use phylogenies in fossils to test how these things connect to broader patterns of speciation. So you might say, what is the relative importance of these factors? What is the rate limiting control or step for successful speciation? So the classic case is to look at reproductive isolation. And the idea is that if some lineages evolve reproductive isolation faster than other lineages, like some clades, clade A, clade B, one clade evolves reproductive isolation quickly, one clade does not. And if reproductive isolation is a key limiting step in the speciation process, then we should see a correlation between the rate at which reproductive isolation evolves and the rate of speciation. And this idea is certainly not new to me. This goes back about 40 years. This is really the ultimate mechanism by which people think generally that sexual selection drives speciation or speciation rates, for example. So, we have clades that might have weak sexual selection and clades that have strong sexual selection. Sexual selection is essentially an engine that drives divergence in mating signals, which leads to faster speciation in groups with lots of sexual selection. And so this is a sort of a direct manifestation of this general idea. Now, back to this assertion here, things that should predict speciation based on sort of this control hierarchy here have consistently failed to do so. So I'm just going to give you a few examples of this. And, one of the things that I think coming into this over the past 15 years, if you didn't know what the results of these tests were, and one of the reasons why I think many of us, including myself, were very excited about this, is that really today, with modern genomic data, phylogenies and the computing power and some of the fancy math that we have and big data, both genomic, ecological, and otherwise, we really have unprecedented ability to test our theoretical and conceptual understanding of speciation. And yet, these processes that should predict speciation rate do so inconsistently or not at all in almost every test where it's been looked at. So a couple of examples here. This one not from our lab. This is looking at rates of pre-mating isolation or song discrimination in birds between fast and slow speciating bird lineages. This is just showing that there's actually 2 curves that completely overlap on that graph, just showing you that the rate of song discrimination in birds, a measure of pre-mating isolation, doesn't the clades that are fast speciating aren't evolving song discrimination faster, as you might predict under classic ways of thinking about speciation mechanisms in birds. Now, that's one example. Here's another. This is from one of my PhD students, Mateus Januario, had a paper on this in AmNat, American Naturalists, earlier this year, where he compiled direct estimates of the intensity of sexual selection from the literature and looked for a number of vertebrate groups and asked how well that predicts speciation rate. Because this is, rather than using sort of one of the things people have used in the past 20 years to look at this question is proxies for sexual selection, like sexual dichromatism in birds, plumage dichromatism as a proxy or an index for sexual selection. Mateus compiled direct estimates where people actually measured the variance in male-specific reproductive success. And what he found was that there was no effect of the strength of sexual selection on the speciation rate for clade. This is actually consistent with the results people have had with proxy variables like sexual dichromatism in birds, but in some ways it's a more direct assessment. This is another example. So this is from an older paper where we directly measured the rate at which pre-mating and post-mating isolation would evolve in different clades of Drosophila, and we found that they were uncorrelated with macroevolutionary rates of speciation. Another example from the same paper where we came up with a way of estimating how fast the rate of post-zygotic isolation, essentially Dubchansky-Mueller type incompatibilities, were evolving within different clades of birds. And so this graph is essentially just showing how the amount of reproductive isolation post-zygotic in terms of hybrid fitness accumulates differently in different clades of birds. So you've got clades like pheasants here on the left, where there's a relatively steep curve showing that they evolve reproductive isolation quickly. And on the right, you've got parrots showing a relatively slow accumulation curve. So if these post-zygotic mechanisms or Dubchansky-Mueller type incompatibilities are a key limiting step for speciation, we would expect clades that evolve quickly in this space, like pheasants, to have faster rates of speciation. So bird clades differ in the rate at which post-zygotic isolation evolves between species, but there is no effect, no relationship between their post-zygotic rate of evolution and their speciation rate. So each of those data points right there is an individual species showing you, how fast it is essentially evolving post-zygotic isolation. So this doesn't predict rates of speciation. Let's look at another key control. Let's think about that first idea from Meyer, this idea of the formation of isolated populations. This is something I was quite optimistic about because it's something I think we can really do a good job of directly measuring. And so we've looked at this in a number of groups now. And one of the ways we approach this problem is to do things like we'll take an animal like this skink from Australia. We take genetic samples from across their range, and we can we can sequence genomes or ddRad data for these things and look at the relationship between geographic distance and genetic distance and plot these out and essentially come up with an estimate of the isolation by distance slope for those populations. And to a first approximation, I think the slope of this line is really a proxy for how quickly these populations are diverging genetically from one another. There's a lot of biology that goes into why that's the case, but one of the things we know is that there are a lot of biological differences that cause different kinds of organisms to differ in these isolation by distance slopes. So for example, these are a couple of South American squamates. You have a viper here on the left with a relatively shallow slope, and you have a microtiid with a relatively steep slope. And so here you have isolation by distance, which is essentially a proxy for population isolation rate. And it varies extensively among species, and it's predicted by some of the biological traits that we expect to predict it. Things that have to do with like dispersal ability, for example. But turns out this propensity for population isolation does not predict at all the rate of speciation, even though we would predict, for example, that things that are diverging more in this space, becoming essentially forming isolated populations more quickly genetically, perhaps because of reduced dispersal capacity, should be speciating at faster rates. But they don't. They don't do it in South American squamates. They don't do it in Australian squamates. And other folks have looked at this in other groups as well. In some cases, a couple of mixed results out there, but I would say the general finding is that there isn't much of an effect. So, another point. Things that should predict biodiversity consistently fail to do so. Let's go back to this heat map for species diversity in marine fishes. What if we could make a similar map of speciation rate to find out where the hot spots of speciation rate are. The classic view, and I expect if you poll, at least up until maybe 10 years ago, if you polled most evolutionary biologists and ecologists, I expect most people would say speciation rates are faster in places where species diversity is the highest. This underlies many classic hypotheses for the latitudinal diversity gradient. Well, we can build that map now, and that map looks like this. In fact, we see the fastest rates of speciation in places where species diversity is in fact the lowest. And you might say, well, maybe this is, the places like the tropical Indo-Pacific are essentially dead zones in terms of, or cold spots in terms of speciation rate, and high latitude, low diversity, polar and high latitude cold water oceans are essentially hotspots for speciation. This result I do not think is a fluke. I would say this is, it's just the relationship between species richness for individual geographic bins and their estimated overall speciation rate. So there's a negative relationship in this case between the rate at which species form and the speciation rate of that geographic region. Well, maybe this result might think maybe this result is just a fluke. Maybe it's just something about marine fishes. But in fact, this result is very general and applies across almost all taxa where it's been looked at. So this is speciation rate in plants. Obviously, plants show a tremendous latitudinal diversity gradient, but there's no correlation between speciation rate and latitude in plants. It's true for other taxa. The paper that was just published in Communications Biology looking at it in amphibians. Despite a huge latitudinal diversity gradient in frogs, there is no significant difference between tropical and temperate lineages in terms of speciation rate. If anything, it's the temperate lineages of frogs that have faster rates of speciation. And the same is true for many other groups where people have looked at this. Another example from ants, strong latitudinal diversity gradient, no effect of latitude on speciation rate. So we might look at other dimensions of this pattern. What about clade patterns? So this is from a paper we published last year. This is looking at a super diverse, famous clade of lizards that Mariana talked about earlier today, the Lyolemines with about 320 species. This is a fast speciating clade and it's high diversity, so maybe there's something there. But you don't have to look very hard to find other counterexamples. So this is the group that we study in my lab. These are Australian sphenomorphine skinks, about the same diversity, and a speciation rate that is only about 25% that of Lyolemines. So we could sort of find examples of fast speciating diverse groups and slow speciating diverse groups. And overall, if you look at sort of family level clades or subfamily level clades of lizards, this is the relationship between speciation rate and species diversity. So there's kind of a weak effect if you include really small clades, but once you're out of that really small clade space, there's no relationship between speciation rate and the diversity of clades. And across the tree of life, one of the things that we have learned is that there's a huge amount of variation in speciation rates. This is just sort of a summary here of speciation rate variation across the tree of life. And it's largely uncorrelated with species diversity. So it's an interesting problem. In some ways, very parallel to another pattern of where there's a lot of variation in genetic variation among taxa that largely appears to be unexplained by life history and other features. But in any event, all right. My final point on this aspect of biodiversity dynamics is that short-term estimates of biodiversity change almost always seem to slow down over long time scales. So this is a very pervasive pattern. It's one that people have been talking about for a while. This is a figure from one of my papers from about 10 years ago, looking at how the ages of clades and speciation rate scale with each other for angiosperm clades. On the right, you have another paper from another figure from a Harmon et al. Review of this topic, looking at relationships between the ages of clades and their estimated macroevolutionary rates of speciation. Again, showing this sort of strong slowdown effect. This is a pattern of bivalve diversity through time in the fossil record. And it appears to show and has been cited to show exponential rise in diversity through time. One of the things that we did was we went to this curve and we said, let's pretend we're an observer sitting here at a different point in time, like the Triassic. So let's pretend we're an observer and let's apply the kinds of methods that we use today in modern macroevolution. Let's apply it to species diversity then and see what predictions it makes about diversity through time. Let's see what happens. We know now with the fossil record what's going to happen, but we can pretend we live in the Triassic. So we'll say we're going to estimate the rate of diversification using similar methods to what we would do today, predict future diversity. This is what you get when you do that for Triassic diversity. So you get massive overestimation of diversity at all future time points. And really there is, while it looks like there is an increase, and to some degree there is obviously a real increase, this is very, very far from exponential. Exponential is really, really, really strong. So this is if you go to the Cretaceous, you'll see the same general thing. So I think this is a very general feature of paleontological diversity curves. And one of the things that this leads to is this artifactual rate scaling, where these are looking at a bunch of paleontological time series. This is just showing that if you looked at these different groups of organisms at different points in time, you would just see the longer the time scale over which you measure rates, the more the rates seem to slow down. So this pattern is not saying that rates are getting slower. This is an artifact from assuming that clade diversity increases exponentially through time in the kinds of models that we use for doing macroevolution. So here's the cynical take-home conclusion from this part of my talk. Are we learning anything at all in macroevolution and macroecology, at least with respect to biodiversity dynamics? So, I don't know, maybe. But actually, I think that nature is telling us something important through these results. I don't think these results are artifacts. I think there's a lot of uncertainty in how we measure rates. There's a lot of uncertainty in how we measure isolation by distance. There are huge uncertainties in all of these things. But I do think the overall thing that they're pointing to is quite compelling. And I think this is telling us something important about how the world works. And I think that we should start listening a little bit more than we are. And I'm going to sketch out now a general conceptual model for how I think we should be thinking about these patterns. And so this is a conceptual and graphical model. And really, this is a model that goes back to one of the famous conceptual models in evolutionary biology and ecology. And this is the MacArthur-Wilson theory of island biogeography. And in this theory, what you have is a dynamic rate of speciation or extinction and immigration going into a community, like an island. So from the original paper in 1963, here we have a per species rate of immigration. So the immigration rate for new species declines as a function of the number of species present in the community. And here we have a per species rate of extinction. Where those lines cross becomes the point where in this dynamic models kind of framework, this is going to generate an equilibrium. There doesn't have to be any connection between speciation and extinction at all. It's just where the lines cross is exactly the point where your input balances your output, or input balances your outflow. And so the equilibrium emerges from dynamic rate curves. I would say that in getting to where we're going to get to with macroevolution, this is one of the most misunderstood curves, I would say, in biology. And one of the reasons for that is that there's a tendency to read into this curve that, there's this equilibrium point and it's sitting right here. There must be something in this model that says there is this carrying capacity for islands. We could only stick K species on the island. This is not how the model works. The model just says if there is, for any reason, if your immigration rate curve drops as a function of species diversity, and if your extinction rate increases as a function of species diversity, even if it is for non-biological reasons. The original model they talked about did not even have biology driving those patterns. The speciation rate dropped for one, or the immigration rate drops for one reason. There's a finite pool of species in the global species pool, and as you put more of them on an island, there are fewer of them available to immigrate. So the rate has to drop. That will give you an equilibrium. There's not even any biology in that. So the equilibrium just emerges from these dynamic rate curves. Now, MacArthur took this idea, and in 1969, in a paper on patterns of community in the tropics, at the very end of this paper, he buries this little figure in the back right here, where he basically shows the macroevolutionary consequences of thinking about this in terms of speciation and extinction, not immigration and extinction for island dynamics. And so, in this original graph is a little hard to parse because he's showing rates for the entire biota. So I'm going to redraw those curves. So I'm going to redraw MacArthur's figure with per species rates. And here you've got the speciation rate declining as a function of species diversity. Here you've got the extinction rate increasing as a function of species diversity. And these curves would predict an equilibrium diversity. Now, what MacArthur was saying was that, hey, maybe these curves differ for tropical and temperate communities, and what we have is something that looks like this. This is our temperate equilibrium, but we have different fundamental curves between speciation and extinction and diversity for tropical communities, and maybe they look more like this. So the per capita effect of changes in species diversity on a tropical curve is different from the per capita effect of a change in species diversity on, or an effect of species diversity on the rate for the temperate curve. So we'll dissect these equilibrium points right here. Now, here you have the different equilibrium values. You've got fewer species in the temperate zone, more species in the tropics. Now, this illustrates some really, really important points that I think are very widely misunderstood. As I've drawn this, the speciation rates, as you would measure them right now in the present day, are exactly the same for the temperate and the tropical zone, despite the fact that you have more diversity in the tropics relative to the temperate zone. So in other words, at equilibrium, the speciation rate for the tropical zone and the tropics and the temperate zone are exactly equal, and the same is true for the extinction rate. Now, for reasons I won't go into, we are actually quite good at estimating the speciation rate and almost abysmally poor at estimating the extinction rate. But we can estimate speciation rates. This can explain some of these patterns we're seeing in terms of lack of coupling between species diversity and speciation rate in a geographic sense. Now, Mike Rosenzweig published a paper in 1975 where he, in great detail, dissected this graph from, the final figure from MacArthur's 1969 paper. And he starts his paper, which is a chapter in this sort of famous volume by Diamond and Cody on the assembly of ecological community. He starts this by saying, in 1969, Robert MacArthur published a graph which indicated without comment. Without comment part, I'm highlighting this to say MacArthur really didn't say that much here. He kind of alluded to this idea in other points after this and in his 1972 book right before he died. But really, Rosenzweig is the one who I think pushed the theory on this, both in this paper and in subsequent work and in his 1995 book on species diversity and ecological communities. If you're interested in this chapter, send me an email. I'm happy to send it to you. It's pretty hard to get, but I could send you a PDF. And so I think about this model not as the MacArthur-Wilson model. I think about this with respect to speciation rates as the MacArthur-Wilson-Rosenzweig model, because I do think Mike Rosenzweig added greatly to the theory that underlies this and what our expectations should be for how this would affect especially large-scale complex biogeographic assemblages of species, like in South America, for example. Now, I have three key take-homes for thinking about this model. First of all, the MacArthur-Wilson-Rosenzweig model, or the MWR model, is an equilibrium model. And that means that biodiversity can be decoupled from the amount of time available or from diversification rates. So if you looked at how species richness would accumulate under this sort of model, starting with a very low species diversity, it would eventually reach some sort of stable state. And that means that the amount of time you've been in a community, or the amount of time a biota has been evolving, isn't going to be predictive of what the species diversity is anymore. Obviously, this is an idealized curve, by the way. There's going to be lots of noise in reality. There's lots of details. But the second point, this is one of the most important points. This model does not care why rates vary. Does not care at all. If the speciation rate declines with diversity for any reason, if the extinction rate increases with diversity for any reason, there will ultimately, at least in theory, be an equilibrium point. I mean, whether we get there or not, or whether we are there or not, may be something we can argue about empirically or based on theoretical expectations of timescales. But the fact of the matter is, this is what is going to happen in the long term. So if I've got, on the left, I've got the extinction rate increasing. On the right, I've got the speciation rate decreasing. Both of these cases are going to give you an equilibrium point. Just to remind you, this is what we usually assume in macroevolution. Evolutionary rates are diversity independent. There is no relationship between evolutionary rates and species diversity. This is the standard birth-death model as applied to, frankly, most of phylogenetic macroevolution anymore. But this model predicts explosive exponential diversity change, which is a pattern that really is almost never observed except at very short timescales. Now, this is sort of where I get to deal with what I think is the biggest misconception about equilibrium dynamics, because equilibrium is a very, very controversial word. You start throwing this around, you generate very, very hostile reactions from people. Now, this is the commonest view of equilibrium dynamics, where species diversity is going to determine the number of empty niches that are available, and speciation rate is going to be a function of how much niche availability you have. So this is sort of the classic carrying capacity idea for equilibrium dynamics. This is where speciation rates decrease as niches get filled, and there is a predetermined number of niches in the system, something like a carrying capacity. This in some ways is really classically how we think about adaptive radiation. Actually, you use the word equilibrium for adaptive radiation, and people will sometimes get upset about it, but at the same time, they are perfectly okay with this basic idea here. So they're holding two contradictory thoughts in their head at the same time. This is the definition of ecological opportunity. Speciation rate slows down as niches get filled. But what I really want to tackle here is this idea. This predicts a scenario like this, where you have, the speciation rate declines. Even if you keep the extinction rate constant, you're going to get some particular equilibrium diversity as a number of niches are filled within the system. This is sort of the carrying capacity model. This can generate equilibrium dynamics, but assuming that it is the only or even most likely mechanism for equilibrium dynamics is a big error. In my view, this is one of the most serious errors that we've made in macroevolution in the last 40 years, because I think it has caused us to throw away an entire line of reasoning about the potential implications of equilibrium thinking in macroevolution. So just to make my point, I'm going to give you a couple of simple examples. Let's get as far away from this idea of empty niches as we possibly can. And I think a great example for this, my sort of counterpoint to a niche-based world, is Hubble's neutral model. So here there is one niche. All individuals, regardless of what their species identity is, use that niche exactly the same way. The interspecific competition is exactly the same as intraspecific competition. There is no niche structure. The dynamics are completely driven by stochastic turnover, individual birth and death, and the probability that individual units that we call species will split into another species. Now, under the Hubble neutral model, because the one thing that it does hold fixed is the total number of individuals that the system contains, like the number of trees in the Amazon basin, you get a relationship between species diversity and population size that looks like this. Species diversity rises, the average size of populations per species has to be lower. It will be lower mathematically. And what that leads to, because population size in this model predicts extinction rate, because ultimately it is a stochastic process, but the bigger your population size is, the less likely or the longer your waiting time is to extinction, you will get this effect right here, where the extinction rate increases with species diversity. More species, smaller per capita population sizes, higher average extinction rate. This is a mathematical reality of this model. And really what it leads to is going the wrong way here. This leads to an equilibrium that is diversity dependent implicitly by this effect of species diversity on population size and ultimately with extinction rate, even though we don't really think about this as a diversity dependent model. Here's another example, and this is from MacArthur's 1972 book. He talks a bit about what regulates species geographic range. And he talks about the role of abiotic factors and biotic factors, where and when those might matter more with respect to latitude. And he was a big advocate of the idea that at least in some places, species interactions have to be important in constraining geographic range. I think this idea has tremendous empirical support. Certainly not the only thing that constrains geographic ranges, but we absolutely know from many studies that competition, for example, and in some cases other interactions like potential for hybridization, cause species range limits or bound species ranges. This is just from a summary paper from about 15 years ago now, but it was looking at the number of studies at that time that were supporting competition as a potential limit for species ranges. And so the majority of cases, there was at least some support for competition between species being an important limit on geographic ranges. Well, what are the macroevolutionary implications of that? The more species that you have in a system, the smaller your average geographic range size will have to be, because at least some of those species are going to push on each other and restrict range expansions, and therefore you're going to have declining range size as a function of total species diversity. More species, smaller average range sizes. And this is probably one of the best supported generalities in conservation biology and macroecology, and that's that range size is consistently one of the strongest predictors of extinction risk. This is just one little example from the fossil record from about 10 years ago, looking at range sizes in different groups of marine mollusks in particular, and brachiopods, and asking whether, to what extent, what is predicting their long-term extinction risk. And it turns out in this case that geographic range size is by far the dominant predictor of extinction risk. Now that result applies both with paleontological data and with neontological data. If we look at like what determines threat status in a species today, it tends to be very highly correlated with geographic range size. The major predictor of extinction risk. But what this leads to is that you have a relationship between range size and diversity, which leads to a relationship between species diversity and extinction rate filtered through this effect of range size. So again, it doesn't matter why the rate of extinction is increasing, it is going to at least have an implicit correlation with species diversity. So you have an automatic vehicle right here for generating an increasing extinction curve with species diversity. So I would say, sort of wrap this part of the talk up, I would say that almost all ecological interactions have some consequence for speciation and extinction at some scale. In fact, I struggle to think of any ecological interactions that don't have some consequence for the shape of those speciation and extinction curves. Any effect of species interactions on abundance, geographic range, or any other demographic property of species is going to lead, I would say, to diversity dependence at the macroevolutionary scale. And while it's not widely appreciated, even the simplest model of community ecology, Hubble's unified neutral theory, is implicitly diversity dependent. And I would also assert that there is no justifiable model in macroevolution that allows speciation and extinction to be independent of other species for anything but extremely short spans of time. All right, so what does this model predict? This model predicts a lot of what we observe. This model predicts a decoupling between speciation rate and species diversity, or at least the potential for decoupling. It makes no prediction either way. It predicts that diversification rates will show time scaling. Absolutely. If you are more or less at equilibrium or loosely constrained by a equilibrium type process, rates are going to slow down with respect to the time scale over which you measure them, which is exactly what we see. And it predicts that microevolutionary controls on speciation are going to have complex relationships, or maybe even no relationship, with the speciation rates that we estimate from phylogenies of the fossil record, which is something that we see in many studies. And of course, there are many, many other lines of evidence, which I'm not going to go through. There are things like, species diversity should be more or less constant for long periods of time. This is, in fact, one of the strongest pieces of evidence in favor of this general type of dynamic. This land plant diversity curve, for example, showing an increase over, 450 million years. What gets lost in summarizing it like that is that there are features of this like nearly 200 million years where land plant diversity basically did not change. How probable is that under a scenario where there's not some kind of broadly balanced speciation and extinction process? Paleobiologists have long recognized this. So if you're a paleobiologist, I am not claiming to invent something new here at all. Charles Marshall says that this is the equivalence between speciation and extinction is supported by, so many studies that we can essentially consider it a third law of paleobiology. The net diversification rate, most of the time, for most organisms, is 0. I would say that, with respect to the rate being 0, again, I would come back to this idea that exponential growth is very, very, very powerful. We underestimate the power of exponential growth. Darwin recognized it. Chapter 3 in The Origin is essentially an ode, one of my favorite chapters in all of science, an ode to the power of exponential growth. And I think we are essentially underestimating just how potent unregulated exponential growth is in macroevolution in general. And finally, there's a number of other predictions we could make here with this from this general model. The model predicts that this MWR model predicts, for example, that ecological variables like area and energy would be better predictors of species diversity than things like time or diversification rates, which they are. So this is just a paper that just came out in Science a couple of weeks ago looking at this idea. But in fact, this idea that area or energy or are predictors of species diversity is something that goes back decades, and there are dozens or hundreds of papers on this topic supporting this general idea. This is the number one criticism that I encounter in thinking about equilibrium models, especially this particular one, which is that the MWR model is a model without a mechanism. I would say my response is, so what? The model does not have a mechanism. That does not mean we should not be searching for the mechanism or thinking about possible mechanisms. We should be very, very interested in how and why speciation and extinction rates vary as a function of ecological processes. That should, in fact, be the key frontier in biodiversity dynamics side of macroevolution for the next 40 years, as far as I'm concerned. However, many scientific theories are useful. They explain and predict natural phenomena even before we understand the mechanisms, the lower level mechanisms of those theories. We often forget this and we're like, you don't have a mechanism. Well, okay, there are some other theories that didn't have mechanisms and yet we made huge progress in understanding the world without a mechanism. For example, the theory of Mendelian genetics. Pre-1950, we were still talking about Mendelian factors that sat on chromosomes, and yet this allowed us to make all sorts of predictions that were completely mystifying to people about the transmission of genetic information and patterns of phenotypes through ancestry. So, and there are other examples as well. Here's Newtonian gravity, which yielded highly accurate predictions for celestial mechanics or ballistics, and yet we literally had no idea why physically separated objects would follow an inverse square law of attraction. And I would argue actually the same is true for general relativity. It's more mathematically elegant, but at the end of the day, we don't know why mass curves space-time, and physicists don't really know why mass curves space-time, even if it's very, very accurate. Plate tectonics, same deal. Theory of plate tectonics predicts major structural features of ocean basins, distribution of volcanism and earthquakes, and so on, distribution of weird fossils that were associated with Gondwana, all with no real understanding of how mantle convection would drive plate movement, which we understand that a lot better now, but certainly for the first 20 or 30 years of plate tectonics as a theory from the 50s on, we really didn't understand it. It was just kind of stopping there. Well, it must be something about mantle convection that moves the plates. But this theory was nonetheless very explanatory. So my last slide is to summarize a few things. So I think one of the implications here of the idea of the world mostly being in an MWR equilibrium most of the time is that we really need to rethink the assumptions and expectations of most macroevolutionary models, at least with respect to biodiversity dynamics. Another implication is that we need to fundamentally rethink how we study speciation. And we need to think about the assumptions that we make about the importance of speciation for biological diversity more generally. Modern speciation theory likes to present itself as being ecological, but the ecology in speciation, for the most part, is very trivial. Ecology drives the evolution of traits associated with reproductive isolation. That is the most boring way, in some ways, that speciation could be driven by ecology. What is completely lacking is any connection to population ecology. But I would say that in thinking about it from this MacArthur-Wilson-Rosenzweig perspective, population ecology becomes the fundamental bridge between microevolution and macroevolution. And I would say at the same time, though, this is both a frontier and a very hard problem. I think we have a lot of challenges in testing specific mechanisms that underlie MacArthur-Wilson-Rosenzweig dynamics. But I don't think that should preclude us from thinking about the explanatory power that model might have for some of the patterns that we are finding, especially some of these mystifying sorts of patterns results we are getting with respect to biodiversity dynamics and macroevolutionary rates. All right. Gracias por escuchar.
¿Ninguna pregunta por acá? Ah, sí. Ahí tenemos una. Yo te alcanzo.
Thanks so much for the talk. I enjoy it very much. Um, In many ways, what you present today remember me parallel in evolutionary biology, which is people start to test rates of evolution in traits. And we have explicit models, for example, oh, this is sexual selection, this is directional selection. We know it is. And we test it, and you expect those rates to be large, right? Wrong. They are very low because you are diluting the change for time in a long time. So you have natural selection operating, you produce a massive divergence, but then you get that amount of evolution and divided by 60 million years is a very low rate. I wonder if it's not the same parallel with speciation rate.
So you mean in terms of basically in terms of the parallel there? I mean, I think the parallels are huge. So in some ways, on a fundamental level, you're really looking at the hallmark of a constrained process.
Yeah.
You're really looking at the hallmark of a constrained process. I mean, obviously the details are going to be extremely different, but from a purely statistical perspective and throwing away biology, you are looking at things that are clearly phenotypic evolution from by natural selection is constrained. Like that's, short-term estimates of selection will drive things, but their, body size just can't go off without bounds, right? So any constrained process is going to give you, so in some ways, it seems to me like such a trivial observation that rates should slow down at this scale. So I guess I think there's strong parallels at that scale. I guess probably the other thing would be that, I mean, I think there's maybe a looser analogy between the ways in which we seek processes at short time scales and seek to extrapolate them over deeper time scales. I think we're doing maybe an okay job of showing that, that's problematic in diversification rates. I mean, people do, it drives in phenotypic evolution. I mean, I have to admit, sorry if this bugs some people, but like, equivalences between drift and Brownian motion, that kind of stuff drives me nuts when people try to do that kind of stuff with phenotypic evolution as well. I don't know, I might be rambling. Is that kind of getting at your question? Oh, okay. Gracias. Well, I might have just spoken too fast for a whole hour. So
yeah, thanks.
She's waiting an hour to tell me that.
¿Hay alguna pregunta en Guatemala? ¿En Guatepeor? Tampoco. ¿Alguna más por acá? No, espera, porque no te va a escuchar. No, espérame porque él avisó que tenía un problema auditivo. No se vayan, que tengo que hacer unos anuncios muy, muy importantes. No dónde es el próximo Clebol, eso todavía no.
Thank you so much. That was great. So can you comment on why you think it took so long to put together, from MacArthur Wilson theory, it's kind of an obvious step. And for me it was like, yeah, obvious. But why it took us so long to make that connection, and why we have been doing so much macroevolutionary research without making that connection?
That is a very complicated problem, but I would say, to me, it comes down to a number of things. I would say, first of all, I think in community ecology, the idea of equilibrium became a very bad word, starting with the sort of null model wars that happened in the 1970s and 1980s over community structure. So there was the Simberloff, Diamond, Strong wars over whether communities were over the role of equilibrium and whether dynamics were chance and so on. I think that left equilibrium with a bad word. And so I think people have been reluctant to entertain equilibrium models. At the same time, I think there has been the prevailing assumption that equilibrium has to entail fixed, deterministic communities with a carrying capacity, with a set number of niches, and species fill the niches. And even the macroevolutionary models in paleobiology that are used are essentially, they are taking carrying capacity models from population biology, like a logistic model, and putting them in the fossil record. And so I think that led to a bit of a, kind of a negative reaction in that sense. Even though I will say, well, Sepkoski, for example, who, essentially developed the Sepkoski curve for biodiversity change, And he was the first person to apply a logistic model to diversity dynamics in the fossil record. But in his paper, he's actually quite careful about talking about the ways in which population extinction could drive these kinds of patterns. So people are using these models like logistic phenomenologically as like, let's just see what we can get with this model. But ecologists and others are interpreting it very literally as like fixed carrying capacities and so on. So I think that's been a problem. And the final point is real, and that's that biodiversity has definitely increased. There is no question that biodiversity has increased. So you can't look at the history of life through time without seeing that diversity has increased, that innovation has happened, and that on some level it absolutely increases. I mean, so my view is much more like, innovations happen and they dramatically expand where those equilibrium points are going to be, but it's happened over and over and over again. When flowering plants evolved, that unquestionably triggered such a massive cascade upwards in terms of terrestrial biodiversity for all other trophic levels. Think about the number of insect species that are host plant specialists. What insect, that alone, trade-offs, host plant specialization with lots of now angiosperm species, each of which is diverging with their own sets of chemical defenses, must have had this colossal effect on insect biodiversity and presumably on everything else. So it's hard to look at the history of life without being impressed by the fact that that has also happened. So we are asking in some ways to hold this more, rather than, life is regulated or not regulated, we have to hold this more nuanced view where innovations happen and absolutely drive increases in diversity and change those set points. But it doesn't mean that most of the time, over like a 20 or a 40 million year time scale, you might see what's essentially, a quasi equilibrium along the lines of that sort of, dynamic rate curve, what you're getting out of dynamic rate curves. So.
¿Hay alguna pregunta virtual?
Thanks, Dan. So from your last point, I think there's an implication I'd like to see whether you agree with. When we are in a certain understanding.
I was trying to back up.
It's okay.
But anyway.
When we're in a quest to understand the evolution of biodiversity, speciation, extinction, I think sometimes we conflate our biological intuition with mathematical realities. So you made a compelling argument that any reason for changes of rates will result in these patterns. That ought to exist in theory for those new pollinators once there's a new opportunity with an angiosperm. Would you agree with that? And so it becomes a layered effect of phenomenological models with new opportunity?
Yeah, I mean, I agree with that. Is from the perspective of, I would say, if I had a whiteboard, I would draw this out because I think about that exact, the example of thinking about angiosperm diversity, for example. I generally think about that curve as really, you can only think about that kind of model as, sort of within a trophic level. And really what you would be looking at from is, changing the evolution of angiosperms from the perspective of the insect curve would have been changing the resource base for that entire trophic level, and it would have fundamentally shifted the relationships between speciation and extinction rates and diversity for beetles, for example. So I don't see a conflict there in that sense. It's just a matter of thinking about the sort of trophic scale at which it applies. So from that perspective, species diversity of angiosperms is facilitating species diversity at another level. But I think within trophic diversities, that's not really happening. You get a change in those curves from this being reset by, the fact that you suddenly have lots of angiosperms. But in general, yeah, I do think that especially cross-trophic diversity shifts, the evolution of entirely novel forms of life are, creating opportunities in other trophic levels, whether it's parasite biodiversity or herbivorous insects or what have you. So it has to be a huge catalyst for the, expanding universe of biodiversity overall.
Bueno, se nos acabó el tiempo para las preguntas, así que cualquier cosa después lo pueden contactar a Dan. Con esto yo primero propongo agradecerle y después, por favor, que no se vayan y les doy los anuncios. Hoy, a las 19:30, hay una caminata bilingüe por el centro histórico. There is a bilingual walk today at 7:30 p.m. So please be here in the door if you want to join. A las 21 horas hay una proyección de una película, Las dos manzanas, que es un encuentro entre Darwin y Rosas en el espacio Inca. Están todos invitados.